So under carbohydrates and lipids, you have bonding properties and functions of lipid, phospholipid bilayer, which are very easy. So coming to carbohydrates, what type of a molecule is a carbohydrate? How will you identify a carbohydrate molecule from the other molecules?
Okay, well,
Hmm.
they are they're like,
Hmm.
for example, okay, carbohydrates are made of carbon, hydrogen, and oxygen,
Yes,
and
correct.
their bonding is covalent between them,
Why
and
is it called as covalent?
because it's two nonmetals that are bonding together.
Okay. Is it that is why do we call any molecule as a covalent? Like covalent bonds since two non-metal bonds together, we call that as a covalent bond. Isn't it something that is
Yeah.
related with sharing of electrons?
Also, I don't know, the
So
way the
share.
easiest way for me to remember it is that it's usually
Hmm.
between two nonmetals and then
Yes,
share an electron.
sharing of electron pair with the other one. So when we talk about a covalent bond, as it is stated, it's a type of bond in which electrons are shared between two neighboring atoms. Then only you go in to talk about metals and non-metals and other state. And we consider that as a stable bond. Why do we consider that as a stable bond?
Because all electrons, there's no loose electron. All electrons are bonded with something else or shared with another atom, which is why it's stable.
Okay. So the outermost electron is completely filled. That's
The
the
outermost
reason they
shell,
gain.
yeah.
Yes, the outermost shells are completely, so they share their outermost electrons and they fill up their complete outer electron shell. That way, that is why we say that the covalent bonds are very stable. And atomic number of carbon.
Atomic number of carbon, I think it's 6.
Okay. So this and all is common thing, no? You should know, you should not think itself. Getting six, again, you should be very fast in answering atomic number of carbon and all. It's very, very simple. Okay, how will you differentiate between a carbohydrate and a fatty acid in its structure? So what type of structure does a carbohydrate differentiate itself or fatty acid differentiate itself from a carbohydrate? We know both are carbon compounds or organic compounds containing carbon, hydrogen, and oxygen. So how will you differentiate a fatty acid or a lipid molecule?
Okay,
Like,
there's way less oxygen in a fatty acid than in a carbohydrate molecule, monosaccharide, for example. And the
no. My question is what makes carbohydrate and fats different or lipids different? How will you differentiate between both? So you're given different structures and you're asked to identify a fatty acid.
Okay, well, one of them would have like a specific, I forgot the name of the group, but there's an additional group at the end of a fatty acid chain.
Carboxyl group. So it has a carboxyl group at the
Yeah,
end.
the COOH group.
Yes, so that is carboxylic group. So that is how, in case of proteins, how will you differentiate? Proteins have what group?
Oh, do they have a phospho group?
Proteins.
Oh, the amino acid group. The
So
amino.
amine group, NH2, right? And so this is a very common question asked. Different structures will be given and you will be asked to identify either what type of a molecule or you'll be asked to identify the molecule itself. In case of nucleic acids,
The R, no, there's the R group.
R
No,
group is in protein.
sorry. It's gonna be I remember the structure. There's the pentose with the thing and the NH
It
and
has a pentose sugar. It has nitrogenous bases added in one time. And so it has a nitrogen containing compound,
yeah,
nitrogenous base, and a
those
phosphate
are
group.
different.
Okay, so that is how you identify a nucleic acid. So in carbohydrates, you call them as saccharides. Saccharides means
Many.
saccharides, just saccharides. I didn't say poly or I didn't say
Oh,
mono.
isn't that like just one unit?
No, saccharides Ides are nothing but simple sugars. Sugars. The term saccharide is sugar.
Oh, okay.
Okay. So saccharide, monosaccharide means it contains only one sugar unit, and if it is a polysaccharide, it contains many sugar units. So glucose is a common example. Structure of glucose is very, very important. And okay. So one glucose unit combines with another glucose unit, and by what type of reaction?
By condensation
So
reaction,
what
there's
is
a one
condensation?
for
Uh,
it's
come
an
again.
anabolic reaction. It's basically the formation of one larger molecule from two smaller molecules, right?
No.
And you get
Where,
a
so this is one
water as
glucose
well.
molecule, which you correct. So this is one. So where does the bond happen between which carbon atom and which carbon atom? That is the
The
most
end carbon
important thing.
atoms,
Amma.
the 1,4 carbon atoms.
1,4. So alpha 1,4 and beta 1,4. What is alpha and beta?
Those are the two different kinds of glucoses where the OH group switches direction on the
So they are
first carbon
isomers.
atom.
Correct. So they are called as isomers. They have similar structures but different properties. And as you told, the hydroxyl group, whatever is you are seeing is what glucose.
Yeah.
What is this glucose? Alpha or
Oh,
beta?
alpha.
Sure.
I think the beta one is the one down.
This is beta glucose.
Also, alpha is the one down.
Yeah, alpha will have the OH down. Okay, in the first
Okay.
carbon atom. So this is beta glucose. Please remember all this. These are important MCQs. Right. And what we were talking, yes. So carbon-carbon bonds, condensation reaction, where there is a removal of water molecule and first carbon atom and fourth carbon atom combined together. So you have polysaccharides where many many sugar units combined together with the same condensation reaction, but it varies from one to another. So carbon, can it bind to non-metallic elements?
What do you mean by non-metallic elements?
Can it form a bond with non-metallic elements, which is not a metal?
Yeah.
Yes, so it has the ability to bind with other carbon atoms or to any other non-metallic elements, and there is single and double bond formation. So
Yeah,
please,
that's only for fats though.
yes. Can you come again?
For fats, that's where the huge difference comes.
Yes, so saturated and unsaturated. So monomers are individual units, polymers are many units together. So we spoke about the condensation reaction, which is so the bond that is formed between the first and the fourth carbon atom is called as alpha
Glycoside.
1,4. Very good. So alpha 1,4 glycosidic bond, which is a covalent bond resulting in the formation of a disaccharide. This formation is very, very important. Many times they've asked us to demonstrate the condensation reaction, showing the removal of water molecules and formation of a glycosidic bond. Okay.
Mm-hmm.
Starch and glycogen are examples of polysaccharide. So opposite of condensation is hydrolysis reaction. What happens in hydrolysis reaction?
That's the breaking of the bond.
Yes. So there is breaking of
Where you use water, where you
very
break a
correct.
polymer into two monomers or a
Correct.
disaccharide into two monosaccharides or yeah.
Okay. So the glycosidic bond is broken by the addition of water molecule. So a disaccharide will be broken down into monosaccharide, and so that 1,4 bond will be broken. That is what is hydrolysis, and these are reversible processes, which is anabolic and catabolic in nature. Right?
Yes.
And so coming to monosaccharides, the common monosaccharides that we learn is glucose, fructose, and galactose. So we have here hexose sugar and pentose sugar. I hope you know what is the hexose and what is pentose, right? And 5
Yeah,
carbon
hexose
sugar
is
and
6 carbons and pentose is 5. Like
very
for
good.
some
So
deoxyribose, it's a pentose sugar.
So you should know, glucose is the most common energy source, and fructose, they are naturally present in fruits, and galactose is present in dairy products. So coming to properties of glucose, we already discussed this. They exhibit in two isomers, as alpha and beta glucose. where you can see the orientation of the OH group different. It's a soluble molecule. Glucose is a soluble molecule. So where does a soluble, so property of this is also an extensive response question. Describe the properties of glucose. So in this case, as why is glucose soluble? What makes it soluble means what will be your answer? It's already there anyhow.
Because when
Due to
when you
which nature? Hmm.
because when you're trying to break the bond, you require water,
No, 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 no.
right? Which
Glucose
makes
is a soluble molecule because of its polarity. So here itself, the solubility of the glucose is due to its polarity. It contains many hydroxyl group, which are polar functional groups.
uh-huh.
And addition, oxygen that is present in the glucose has a partial negative charge and carbon has a partial positive charge, giving them polarity. So that nature enables them to be in the soluble form.
Oh, okay.
Apart from this, as glucose is in equilibrium with an open chain form. Have you seen the open chain structure of a glucose?
No.
No? Hmm. Like open chain structure is not that. It will be like this. Hmm. Can you see the one here? The middle one.
Oh.
This is the open chain structure. So why does it why does it become open chain? Is because the C1, the carbon one, no, has the ability to rotate, and that's the reason it becomes open at certain point of time. As a result, it is also considered to be a soluble molecule. Because of this rotation only, there is formation of isomers, which is alpha and beta.
Right, because in one direction it'll be alpha, and the other direction will become beta because of the OH group.
Yes. So since glucose is in equilibrium with the open chain form, carbon 1 has the ability to rotate and alpha and beta randomly assigned. So major cause of glucose molecule being soluble is its polarity. Because of that, where and all it is applied, application of soluble glucose or glucose being soluble. Where and all is that property being used up?
For example, when we eat it and our body digests it, it has to
Okay. The
blood.
common, yes, correct. And transported in the blood. It can dissolve in plasma. Okay.
Yeah.
And the hydroxyl group of glucose can form hydrogen bond with water in the plasma, enabling them to get transported. So glucose is needed for respiration, cellular respiration, aerobic respiration, correct?
Mm-hmm.
So that is the second. So why reasoning out is very important. Why is glucose stable? As why is glucose stable? Why is glucose soluble? Means we should be able to reason out. Similarly, glucose is a stable molecule. It's a cyclic molecule because you can see hydroxyl group situated in the axial region. What is axial region? What is axial?
I don't know.
Axis means
Axis.
uh,
Uh, I guess yeah. How to
that
explain that?
90 degrees axis, my XY.
Yeah, exactly.
So that is what we call this as this is a cyclic molecule with OH group situated in axial regions of the molecule. So axial bonds of the glucose are bonds that are present in 90-degree place with the ring. That is what we call it as axial region. So this is what that makes the glucose a stable molecule. So because of its stable nature, how it is being used up? It is being used up as a storage, energy giving storage substance, and energy giving, structure giving, stability giving substance in the plants, and starch and glycogen in the storage of glucose in plants and animals. So all these are its applications, glucose being a stable molecule. Glucose can be oxidized. What is oxidation?
Would you? It's when
Hmm.
oxidation is like the addition of glucose, or the loss of electrons.
Okay, so here oxidation, the thing which you're able to see is the oxidation of glucose, the reaction that you're able to see. Glucose is getting oxidized in the presence of oxygen. So this is a
Yeah.
very important reaction, correct? And am I right? And so this is a very important reaction in terms of cellular respiration. So it is addition of oxygen to a molecule and removal of hydrogen. Am I right?
Yes.
Correct? Or loss of electrons. And so oxidation plays so it's an oxidizing molecule. So that is a fourth property of a glucose. And we need to say where it is applied. So oxidation of glucose plays very important role in cellular respiration. This is about properties of glucose. Any clarification needed here?
For now, no.
Okay. Next, polysaccharides, talking about starch. So starch is a polysaccharide. It is made up of two different and polysaccharide. One is amylose and amylopectin. Amylose and amylopectin. So when you take the structure of an amylose, this is amylose and amylopectin. Common question asked, compare and contrast amylose and amylopectin. So how will
Oh
you
my God, I
compare
love
and
this question.
okay.
I had it in the past two tests for Team B, and I always got full marks on it. So
So
basically, both of them are made from alpha D glucose, right? Then starch has alpha 1,4 and alpha 1,6 glycosidic linkages, and so does amylopectin, right? Then another thing for their
Correct.
structure
So I think you are
is
very familiar with this. Yes. So
yes,
as both are polysaccharide, I think there's some problem. Right? That can be a compare. It is alpha amylose is alpha 1,4 glycosidic and amylopectin is alpha 1,4 and alpha 1,6. That's the reason they are branched. And it has a
yeah,
more complex dimensional thing. And
yeah,
talking
it's
about
insoluble.
yes, but amylo content, if you see only 20%, but amylopectin constitute about 80 to 85%. And so when you see amylose is less soluble in water, amylopectin is more soluble in water. So these can be taken as some of the compare and contrast questions, which will help us. Is right. So we know
Yes.
starch. So coming to the main element of starch. So starch is a main storage compound in plants, and we know it is stored in seeds and roots, and it has a as so. But these separate that together, like both amylose and amylopectin, is insoluble in water because it's of large size. But that is the one that helps them maintain the osmotic balance, and that
Yes.
is we need to know. And it has to be broken down in order to provide energy. The next one is glycogen. So how will you differentiate starch and glycogen?
Oh, okay. So basically, they both have the same monomers, alpha D glucose, and the linkages are
Mm,
alpha 1,4 and alpha 1,6 glycosidic
mhm,
linkages. That, but no, wait. Then
mm,
they're both coiled. They're both like having a
mm,
core structure, and they're both branch structures, as
mm,
well as they're both compact and insoluble. Like they have most of the same
mm.
structures as well, but just starch is a form of energy storage in plants, and glycogen is an energy store in animals. That's the
Mm.
main difference between them.
Okay, so yes, so this when we talk about glycogen, as you told, that is in a stored form in plants, and this is a stored form in animals, and you can see them stored in liver and muscle cells. And this is also glucose molecules that is linked together through both alpha 1,4 and alpha 1,6 glycosidic bond. But frequently you have alpha 1,6 glycosidic bond that occurs for every 8 to 10 or 8 to 12 glucose molecules. It's a highly branched structure. Similar
Mhm.
to starch, this is also insoluble because of its molecular side, but helps in maintaining the osmotic balance. And liver can break down glycogen, and once they break down, they have been released into the bloodstream when your glucose level drops.
Yeah,
Glucagon
that's
hormone, correct?
insulin, right?
Yes. So insulin is when it is more, glucagon when
Oh,
it is
so
less. So
glucagon.
that
Okay.
glucagon will yes, glucagon will break down the glycogen which is present in the liver, and it will take part
Be
in the
to
reaction.
blood.
That yes. So that is about starch. Next, moving on to cellulose. So cellulose means the first thing we remember is they are plant cell wall. Correct. The cell wall, plant cell wall is made up of cellulose, and they are made up of beta glucose molecules. Also,
Yeah, and
yes.
they are straight
Um,
unbranched, go through beta 1,4 glycosidic linkage. Then they are straight
yes.
unbranched change, and the
Correct.
orientation of the beta glucose monomers, they alternate, right? So they're linked
Mhm.
180 degrees from each other. They
Right.
the chains, they're cross-linked or something like that by hydrogen bonding, and these form like cellular fibers which are really strong and elastic, and they make the plant cell wall and give rigidity and support to the plants.
So, right. So, you have already explained everything. So, this structure is also very important. And so, you can see the structure over here. So, they are arranged in microfibrils. Like you told me, right? Long unbranched chain. So, they group together to form a structure called as microfibrils. And these microfibrils are held together by hydrogen bonding. And it is present and between adjacent cellulose molecules. So, that is how cellulose is arranged in plant cell wall. This is because of this microfibril hydrogen bonding, and that molecule becomes stable, and that is the one that provides the property of rigidity and stability for the plant cell, right? And so the tensile strength of the plant cell comes from this cellulose. But this plant cell wall, which we are talking about, is not only made up of cellulose. In addition, they have some protein and lipids, which is helping them in maintaining the shape and integrity of the plant. And so you know how an animal cell is without a cell wall. All it is little not in shape, and it cannot withstand osmosis, and they may collapse under certain cases by there. You would have learned about hypertonic conditions and all, right?
Yeah,
Have you?
hypertonic is when there's
So
like too much salt, hypotonic when there's not.
correct.
Tonic is
Yes. So in that case, there might be collapse in the cell wall if there is no cellulose being present. Next is glycoproteins. So what are glycoproteins?
amino acids.
Glycoprotein.
Oh, glycoproteins, these are carbohydrate, these are like carbohydrate chains that are attached to like certain proteins that are
Yes,
embedded in
correct.
the
So,
phospholipid bilayer.
correct. So they are either branched or linear chains, and they attach themselves with either amino acid or a protein structure, and they are present in many cellular structures. So glycoproteins play an important role in cell signaling and communication. Where have you heard about glycoproteins?
I've heard of them on the surface of membranes, for
Very
example,
good.
especially in like the different blood types,
Very
right?
nice. Good. Right, right. So we would have heard like any signaling, neural signaling or hormonal signaling. They go and bind to the receptors. So
Yes.
that glycoprotein are the one that acts as a receptor. For example, insulin. Insulin binds to the glycoprotein receptors, and it initiates a series of reaction that leads to the absorption of glucose. So this is one of the important roles. Second role is cell to cell recognition. In case of foreign substance entering a body, at that time they recognize, right? So the cells recognize as the foreign substance as something new only by the glycoproteins that is present on the surface. Then ligands, and you would have heard in I think neural signaling. So these glycoproteins
Mhm.
are specific ligand like structures that help in initiating signaling pathways and structural support. In plasma membrane, you have some glycoproteins attached over there. So all that is to provide the structural integrity of the cell. So these are the four different functions of glycoproteins. And ABO blood group is a very good example about what is this? Glycoprotein. So what is the theory behind that ABO blood group?
Oh, you're asking me?
Yeah, yeah.
Oh,
What
okay. So
happens there?
basically,
How is that
um,
recognition, receptors and all we are connecting over here in this application of ABO blood group?
blood group A has one kind of receptors or one kind of
Mm.
glycoprotein attached to the surface, to the membrane surface. Blood group B
Mhm.
is different, A and B has both, and blood group O has none, which is why it's the universal blood donor.
So here you can see all those surrounding the blood group, right? Those are all the surface glycoprotein molecules. So here you have in the incompatible. So when a A blood group receives B, what happens?
Basically, they won't be able to recognize each other, and they're probably going to think it's some like parasite or some other foreign substance, so your immune system is going to try and kill it or try and just get rid of it from your body, and then you're gonna die.
It is called as incompatibility. Either it results in organ failure or death. Okay, it is recognized as some new substance. and
Yeah.
it just like that fades away. So this is about carbohydrates. I hope everything is clear in carbohydrates, right?
Yes.
Okay. Next, coming to lipids, the same way lipids. So when we think about lipids, what are the properties that comes to our mind?
That they're like hydrophobic,
Uh,
like insoluble. They don't like water. Then phospholipid bilayer and like the
mm-hmm,
triglycerides.
mm-hmm.
Then I mean the lipids can either be saturated or unsaturated depending if they're carbon singular double bonds.
Mm-hmm. Yes, so correct, as you
That's it.
said. Yeah, yeah, yeah. That's the thing. So they are hydrophobic in nature. They are water haters. They are non-polar solvents. They are not polar solvents, and they cannot dissolve in water. So because of their non-polarity only, you can see them in the plasma membrane. So phospholipid bilayer, correct? And why is it called as triglycerides?
There's three glycerol molecules.
Yes, so three glycerol molecules. How are they linked together? By
Also
what
by
type of
glycoside,
bond? Here also glycosidic.
but they're called there's special ones. There's something with an E. They're called E bonds.
Ester,
Called,
ester.
yeah, ester bonds between the
Yeah, right. So triglycerides are foods that you consume and synthesized by the liver. For example, butter, ghee, and all they are saying, and they're typically solid at room temperature, and oils which are liquid. You have another type of liquid which is called as wax. So these are water insoluble, they have a high melting point, and they are centrally solid at room temperature, and you know where and all wax is present, and what
the
is the function of a wax?
I don't know. We never really talked about wax in school.
Okay, so wax is a solid form of, it's another type of lipid. So it is a solid, insoluble substance or a lipid that has a high melting point, and at room temperature, it's generally solid. So where it is waxy layer of a leaf, have you heard about it? The
Yeah,
wax,
that's the outer
wax
side.
coating.
Uh-huh.
Yes. So what is the purpose? So that is a lipid. So what is the purpose of that wax?
Uh, for plants, I'm not too sure, but doesn't it like
Prevent
prevent
transpiration. To
and
prevent trans.
yeah, and it makes sure that like water just doesn't leave the
Yes, that is evaporation or transpiration. So it is like a protective layer that prevents loss of water from the plants. So you have triglycerides. Under triglycerides, you have solid fats, liquid oils. Else another type of wax is lipids and steroids. Steroids are,
oh hormones.
yes, so they are also lipids, right? And so you should be able to know the four different forms of this lipids which we are talking about.
So
And
that's
so,
triglycerides. So they're
yeah,
triglycerides, steroids, wax, and
triglycerides, oil,
oh
wax,
oil, wax, and steroid.
and steroids. Yes, correct.
Okay.
So triglyceride, you already told me three glycerol units. So it is also condensation reaction and it is formation of ester bond. So the bond that is formed between I think this is the fatty acid and the glycerol molecule. So removal of water molecule, right? So when three
Yeah.
fatty acid join together with the glycerol molecule, removal of water resulting in the
Water.
formation of, yes, resulting in the formation of ester bond, it is called as triglyceride lipid molecule. So phospholipids, what are phospholipids? It's also a type of lipid.
Yeah,
How?
but instead of two fatty acid chains, you have
Correct.
instead of
So
three,
one,
you have two
mm,
and one phosphate group.
correct.
And this
So
is what
two,
makes the phospholipid makes like one half
yes,
of the phospholipid bilayer structure.
correct, correct. So this is the one that you observe in, and this is the one you observe in the plasma membrane, as you told, and the plasma membrane is both hydrophilic and hydrophobic in nature because of this phospholipid
They're
bilayer.
amphipathic.
Yes, amphipathic. So coming to saturated and unsaturated. And what is saturated and unsaturated?
So unsaturated, they can be monounsaturated or monounsaturated or
Hmm.
polyunsaturated. And that's when
Hmm.
they have like maybe one double bond is monounsaturated and multiple double bonds is polyunsaturated. But if there's
Hmm.
only single bonds throughout the whole thing, that is saturated.
Correct. And
Really?
yeah, you correct only no. And you're very fast. Okay. They are saturated, monounsaturated, and polyunsaturated fatty acid. Very difficult questions and all will not be asked. You will be asked to identify, or you will be asked to select from one whether it is saturated or unsaturated. So we know that this lipids which we are talking about is a very long hydrocarbon chain, and that makes up the backbone of a fatty acid molecule. So saturated fatty acid, there are no double bonds, and unsaturated fatty acid, there are double bonds. Depending upon the number of double bonds, you name them as mono and polyunsaturated fatty acid. Correct? So there is a difference in identifying this saturated and unsaturated fatty acid in structure. How does it look like? Can you see?
The unsaturated ones, they have kinks in them because of the double bonds. But
Yes. So
saturated
due
ones
to
are
the, so where correct. So wherever they have a double bond, there is a kind of a small bend or a king. And why is it so?
because it makes them either liquid in room temperature or solid in room temperature.
No, these kings are like to come close together. So they prevent them from coming close together, so that they can result in liquid at room temperature. These kings prevents the fatty acid molecule from packing close together, resulting in a liquid at room temperature. That's the reason that small bend
Oh,
which we are talking about happens. Okay. So we should be able to know some examples to identify in MCQs. For example, meat, beef, pork, and all is saturated fatty acid. And so they are solids at room temperature. Coming to polyunsaturated fatty acid, all the linoleic acid, then linoleic oil,
all
sorry,
oils.
soya. Yes, most of the oil, salmon, and all will come under that. And so we should be able to have some examples else remembered. So this is saturated and unsaturated. So degree of unsaturation of fatty acid. So how will you determine the degree? So I say that saturated fatty acid have a higher melting point. Why?
Wait, what's the question?
How will you say that saturated fatty acid have a higher melting point?
They saturated have a high melting point because first of all they are solid at room temperature, which means that their
Hmm.
atoms are close together in a
So they
like.
are close together, so making it hard to break. So that's the reason from solid to liquid conversion is very high. They have a high melting point. On the other hand, if there are more double bonds,
If there are more double bonds, that means it's making it more unsaturated, meaning it's gonna be liquid at room temperature, meaning that the bonds are gonna be weaker because the double bonds prevent them from being as closely packed together as unsaturated.
correct. So more the double bond, more the melting point. Correct?
Yeah,
Or no?
yeah.
Why?
Because of
More
room
double
temperature.
bond, more the melting point. How it will happen?
No, more double bond, less melting point.
I told opposite. I think you didn't listen. Okay, fine. So more the double bond, higher the melting point. Okay, so we should remember that. So here also you have cis and trans. What are cis and so same way isomers. So you should be able to identify them. When you have the hydrogen atoms towards opposite sides, you call it as trans. When it is on the same line, you call it as cis.
Mm-hmm.
Is it fine? This identification is also very important. When they are located on the same side, you call it as cis, and when you are located on the other side, opposite sides, you call it as trans. So saturated, unsaturated, mono, poly, cis, and trans. Okay. So coming to lipid, what is the function of lipid?
It helps with buoyancy to keep heat, helps growth and repair of muscles, and it's another form of energy storage.
So it's a form of energy storage. So they're here, they have used the term called as, so it is stored fat in germinating seed for it to grow until they completely grow into a plant. So in form of oil or solid fats, they are stored in animals. So endotherms. So endotherms are, you told me, right? To provide constant supply
Mammals,
of energy. Yes.
warm-blooded
And
animals.
yes, so their fats are stored in the adipose tissue and used up so adipocytes. as liquid droplets and can be broken down into ATP. So that's how they serve as an
For
endotherms.
example,
So
when you don't
insulation.
eat, so is that what
Yeah,
happens? Like, for example, if you don't eat for like ages, then your body basically starts eating itself because it's gonna take like the fats from your tissues that are in
correct, correct. Right. Okay, so this is thermal insulation is your body providing heat. For example, the blubber that is present in your lower grade, you would have
yeah, and
learned
the little
about the whales.
seals.
Yes, so the blubber that is present in the whales, the polar bear, and so you have a thick layer of fat under the skin, even yourself. So that is all to prevent the cold temperature, and it also serves as a form of energy. So this is another application of lipids. So the next one is phospholipid bilayer. So we have already discussed it is amphipathic in nature. So hydrophilic phosphate group and hydrophobic lipid. So how are they arranged? Why is it called as bilayer?
Because they're like facing opposite each other. For example, there are two heads on the outside and in the middle there are
Correct.
like
So hydrophilic
fatty
head and
acid chains.
hydrophobic. Correct. So hydrophilic head and hydrophobic tail is what we say. So they are
It's
amphipathic
the other
as
way
we.
around, hydrophilic head and hydrophobic tail, man.
Oh, I told oppositely. Okay, hydrophilic head, hydrophilic phosphate head, and hydrophobic lipid
Fatty
tail.
acid.
Right, and yes, so this is how they are structured. Diagram is also sometimes asked to draw, enabling identifying all the regions. So when you place them in water, what happens when you face a phospholipid bilayer in water? What happens?
Nothing happens. Water doesn't pass
Nothing.
through.
Does it get completely? What happens when a phospholipid layer is placed in water?
Is the whole thing submerged in water?
Why?
Or what do
Why
you
does
mean?
it want to submerge?
Because
Why does it
because
get
when you say
submerged
water,
in
I'm thinking about the fact that this
it?
this creates the membrane, so half of it is in the intracellular space
That
with
is
the
how only in water, when you place phospholipid in water, it spontaneously forms phospholipid bilayer.
yeah.
That is what I asked. When
Oh,
when a phospholipid molecule is placed in water, it spontaneously forms phospholipid bilayer. That is
yeah,
how the
because the
formation of the
tails are
bilayer.
hydrophobic.
Yes, correct. Next is non-polar steroids have the ability to pass through the phospholipid bilayer. How do they pass through the phospholipid bilayer? How do non-polar steroids pass through this
Through
bilayer?
through like through maybe
Hmm.
protein channels or protein pumps
Hmm.
and stuff like that. Because first of all, they're insoluble, so they can't just pass
Hmm.
through, right?
Hmm. Correct.
So they have
Steroids are, so it's already told they are hormones, which you already told me, right? So it is required by a cell in order to perform certain function. For example, testosterone is required. Testosterone should reach a testis in order to produce sperm. So
Mhm.
that cell will be made up of phospholipid bilayer. So how does it get passed? It gets passed due to the hydrophobic nature of the phospholipid layer. And as you told, it has additional protein channels that supports them to make their passage into it.
Mhm.
Okay. Then cholesterol. You could see some small molecule of cholesterol in the phospholipid bilayer in the diagram.
Yes.
So that is also one of the reason that provides stability and flexibility for such molecules to pass through.
Because some already embedded in the bilayer.
Correct. Okay, so this is about carbohydrates and lipids.